Hello everyone, I am Vaishnavi Agarwal. I will be discussing my master's research project titled Choosing the Perfect Home in Analysis of Real Estate Preferences in Shell-Dwelling Cichlids from Lake Tanganyika at Max Planck Institute for Biological Intelligence in Martinsried, Germany. My work is focused on the shell-dwelling cichlid species Lamprologus ocellatus (LO) found in Lake Tanganyika. These fascinating fish are obligate shell dwellers, meaning that they exclusively breed in nests that they build out of abandoned snail shells. The video shows the nest building sequence by LO, where they place the shell vertically and then cover it with sand, leaving the aperture uncovered for entrance. So obviously, shells are a key resource for these fish, and we want to understand which features are important for the fish to choose their preferred shell. In a previous study, Bose and colleagues demonstrated that Lamprologus multifasciatus, a closely related cichlid species, shows a preference for exaggerated features and highly intact shells using 3D printed models to simulate these characteristics. In the first part of my project, I not only test external features such as decreasing or increasing the overall shell size, I am also testing internal manipulations that involve exchanging the spiral of the control shell with the straight tunnel or by creating a void inside the control shell, leading to three choices for internal manipulations: control, tunnel, and hollow shell. In order to perform the decision task, I built an experimental setup where all three choices for either external or internal manipulations could be presented in two different rounds. Experimental group includes 10 female fish experienced with building the control shell and are recorded for 2 days. An object detection tool named YOLO was used to analyze and track the fish and the three shells over the time course of the whole experiment. Observations that were made in this first set of experiments lead to the curiosity to understand the relation between body size and preferred shell size, which will be examined in the second part of my project. To address this along with Swantje Grätsch, I set up an experiment with shells in various sizes ranging from 2.5 to 4 cm with 3 shells for each size. We then introduced 10 LO at the age of 1 month and performed long-term observations for 5 months. Now when the biggest shell was occupied, I removed the smallest shell row except one shell and then shifted every row ahead and finally added a new row with an increased size of 5 cm at the end. In the preference task, I found that for externally manipulated shells, the small shell was rejected by all individuals and never chosen for nest building or resting during the night. When given the choice between the three internally manipulated shells, I observed a strong preference towards the control shell. In fact, neither the tunnel shell nor the hollow shell were chosen to perform nest building or resting. The results for examining the correlation between body size and shell size found that the small-sized shells were occupied first, and as the individuals grew, they left the small shells and moved on to the larger shells. In the summary graph on the right, we can see that smaller LO were present at smaller shells and bigger LO prefer big shells. Next to these findings, my daily observations shows that 2 months old LO started to manipulate small shells and build a proper nest at around 3 months. This important discovery demonstrates that juvenile and thus sexually immature LO can build nests when provided with an appropriately sized shell. At 4 months old, they show territorial behavior and also build bigger shells, control size. Finally, they learned how to block or hide the shells at 4.5 months, and at 5 months, I noticed a fertilized clutch for the first time. Taken together, these findings let us ask which underlying mechanisms play a role in shell preference and choice. Are LO relying on visual cues or other sensory modalities, such as mechanosensory cues, for example? Understanding this could provide important insights into their behavioral ecology. Furthermore, we made an exciting advancement by capturing the first brain images of juvenile LO. In the future, this approach opens avenues to investigate active neurons and their neurotransmitter profiles during specific behaviors such as nest building or choice tasks by combining third generation in situ hybridization chain reaction with confocal microscopy. Thank you so much for your attention.